Today we'll be discussing about the topic difference between gram-positive and gram-negative bacteria. Okay, so let's see what is bacteria. Bacteria is an ubiquitous microorganism. It means that it is present everywhere. It is both unicellular and prokaryotic. Prokaryotic means it lacks true nucleus, and unicellular means it is a single-celled organisms. Okay, so the simple structure of bacteria will be composed of cell wall, capsule, DNA, pili, flagellum, cytoplasm, and ribosomes. The genetic material of the bacteria will be present in the cytoplasm and not in the nucleus. As bacteria is prokaryotic, the genetic material of the bacteria will be present in the cytoplasm and not in the nucleus. Okay, so based on the classification of gram staining technology or method, the bacteria will be classified into gram positive bacteria and gram negative bacteria. Okay, so based on the cell wall composition, these bacteria will be of two classification, that is gram positive and gram negative. So for finding that differentiation, we'll be using a technique called gram staining. So in gram staining, we'll be using different stains to differentiate these bacteria. Now let's see an overview about gram staining. Gram staining is a process where we can differentiate gram positive and gram negative bacteria based on the different stains used in this process. Okay, so gram staining was first introduced by a scientist called Christian Gram. He firstly introduced this process. Okay, so this staining method helps to differentiate bacteria based on the cell wall. So based on the cell wall, this staining process helps to identify whether it is a gram positive or gram negative bacteria. Okay, so cell wall will be different in gram positive and gram negative bacteria. So keep in mind that cell wall is very important compound to find or differentiate whether it is a gram positive or gram negative bacteria. Okay, now the cell wall is made up of peptidoglycan layer. So it is made up of peptidoglycan layer. Okay, now let's see about the difference, or let's differentiate this gram positive and gram negative bacteria based on their composition. Okay, so now let's see about their cell wall composition. Cell wall in gram positive and gram negative bacteria differs. Okay, so based on the cell wall composition, we can differentiate either it is a gram positive or gram negative bacteria. So if we see in the case of cell wall, it is single layered and smooth cell wall in gram positive, but in gram negative, it is double layered and wavy cell wall. If you look at the diagram, you can understand very clearly. See, in gram positive, this is a gram positive bacteria cell wall, and see here it will be of single layered and smooth cell wall. Okay, so this is the cell wall layer, so it will be a single layer and smooth. But as you see in the gram negative bacteria, you could see that it is made up of double layered cell wall and wavy cell wall will be present. This is a gram negative bacteria, so double layered cell wall is made. Okay, so as you see the thickness, the cell wall's thickness will be thick comparatively. Gram positive will be thick comparatively gram negative bacteria. Okay, so the thickness of a cell wall in gram positive bacteria will be 20 to 80 nanometer thickness, and in gram negative it will be thin. Okay, so it will be thin, like it will be 8 to 10 nanometer. Okay, now let's see about the peptidoglycan layer. So in gram positive, you can see the peptidoglycan layer is multi-layered. So this is a gram positive bacteria. This purple color demonstrates that it is a peptidoglycan layer. So it is multi-layered in gram positive, and in gram negative, it will be a single layered one. Okay, so in gram negative, peptidoglycan layer is a single layer, but in gram positive, it is made up of multi-layer. Okay, now theoic acid will be present in gram positive and absent in gram negative. Okay, now the outer membrane is absent in gram positive. Gram positive bacteria will not have an outer membrane. So if you see in gram negative bacteria, you can see here cell membrane present over here. Okay, so same way lipopolysaccharides is present in gram negative and absent in gram positive. Okay, now the morphology will be a cockeye shaped spore forming rods will be present in gram positive. Spore forming means it will be pathogenic. When it comes under a favorable condition, it will turn as a pathogen. Okay, so pathogenic causes will occur in gram positive, and in gram negative it is non spore forming rods. Okay, now we have seen about the lipopolysaccharide. Lipopolysaccharide is present in gram negative and absent in gram positive bacteria. And if you see the toxin producing, both the bacteria produces the toxin. Which type of toxin it produces matters. Okay, so in gram positive bacteria it produces exotoxin, and in gram negative it produces endotoxin, both endotoxin and exotoxin. Okay, now let's see about the resistance to antibodies. How this gram positive and gram negative is resistant to antibody means, in gram positive it will be more susceptible, but in gram negative it is more resistant. Okay, that is the difference between gram positive and gram negative bacteria. Now let's see about the examples. What are the examples we can differentiate, or what are the microorganisms comes under gram positive and gram negative. Okay, so gram positive bacteria will be staphylococcus and streptococcus, but in gram negative escherichia and salmonella. So this is the basic organism where we can find whether it is in gram positive or gram negative. Okay, after staining process we can identify this is the organism, like that. Okay, so next we'll be seeing about the overview on gram staining. Okay, now if you see the diagram, so here the gram positive bacteria after staining process, if you we can find under microscope that the gram positive bacteria retain the crystal violet color. So that is a type of stain. So it retains, gram positive bacteria alone retains the violet color and stains purple. Okay, so gram positive bacteria will be purple, and gram negative bacteria will lose its crystal violet color after adding counter stain that is called as safranin and alcohol, that is considered to be a decolorizer. So if bacteria is gram negative, then after staining it will appear in red or pink color. Okay, so the cell wall is impenetrable. See, the gram negative cell wall is impenetrable, so that the antibodies is resistant in gram negative bacteria. Okay, so both the gram positive and gram negative bacteria are classified based on their ability to hold the gram stain. So based on the capacity of holding the stains, it is classified, it is differentiated whether it is gram negative bacteria or gram positive bacteria. If the bacteria is gram positive, it will retain in purple color, but if the organism is negative, then it will retain in pink color. Okay, so that is a difference. Now, as we discussed earlier, this gram positive bacteria has multiple layer of peptidoglycan. So that structure makes it rigid, makes the cell rigid. Okay, so since it has a peptidoglycan layer made up of multiple layers, it makes the cell very rigid. If you see in the case of gram negative bacteria, only a thin layer of peptidoglycan is found. Okay, so apart from that, it has an outer membrane made up of lipoproteins, phospholipids and lipopolysaccharides. Okay, so it is made up of lipoproteins, phospholipids and lipopolysaccharides. Okay, so in gram negative bacteria also you could see that the peptidoglycan layer is between the outer membrane and the perisplasmic space. So this is considered to be a perisplasmic space where the peptidoglycan layer is found. So we can see that a peptidoglycan layer in gram negative bacteria lies between the outer membrane and perisplasmic space. Okay, so both the bacteria has the protein. So this is the protein. So both the bacteria has the protein so that it is helpful in transporting the molecules. Okay, so now let's let's recall what we have learned. So this is a gram positive bacteria, this is gram negative bacteria. Gram positive bacteria has a multiple layer of peptidoglycan. In gram negative bacteria, only a single layer or thin layer of peptidoglycan is found. It has protein. Both the bacteria has the protein so that it helps in the transporting molecules. Lipopolysaccharides is found in gram negative and absent in gram positive bacteria. Okay, so the outer membrane here is the peptidoglycan layer, and here it is cell membrane made up of lipopolysaccharides and phospholipids. Okay, so both has a perisplasmic space, but in gram negative bacteria the peptidoglycan layer lies in between the cell membrane and perisplasmic space. Okay, and both the bacteria has the cytoplasmic membrane. The down layer is said to be cytoplasmic membrane. Okay, so after staining, the gram positive bacteria appears to be in purple color. Gram negative bacteria will appear in red or pink color. Okay, so gram positive bacteria will retain the stain, and gram negative bacteria will not retain crystal violet. Okay, that is the difference between gram positive and gram negative bacteria.